I'd like to welcome Jim Giovannoni, who's from Cornell University. Jim, over to you.
It's a real pleasure. I thank the organizers for the invitation. It's an honor to be here. This is my home on Saturday, so it's really nice to get out. So thank you very much. The northern New York's a little cool still this time of year. What I'll talk about today is, I was asked to talk about biotechnology, but I'll put it in the context of horticultural crops, because obviously that's of interest here. And a little bit about, not so much, I think when we think of biotechnology, we think of genetically modified organisms, but I'll bring it back to the tools of biotechnology and genomics enabling us to understand genetic diversity much better in a way where we can take advantage of it and use it often as a solution to some of the things we try to do with more kind of standard biotechnology. So my talk is probably not nearly as interesting as the last talks. I'm sure it won't be. So I'll spend a little time just introducing some things, including my lab. And this is when we put our tomato field in last June, and I apologize, I will mispronounce tomato for the entire talk, so forgive me, please. But you'll notice up here on top, we do it in June, so we are in kind of a cool part of the state. Before that, we could get frosts. And some of the other people in the lab who were not there that day, and this is a student who shared with another faculty, so I just show half her picture. Okay, if you're not familiar with where Cornell University is, and probably everyone is familiar with New York City, we're kind of in the center of the state, very lovely area, particularly during the summer, rural area in the middle of agricultural production, at the end of a very nice lake. My laboratory is actually in a private nonprofit research institute called the Boyce-Thompson Institute, which was established by a gentleman named Colonel Boyce-Thompson, who's an industrialist who actually made a lot of his money purchasing land from farmers going out of business after the Dust Bowl. And he wanted to use his fortune to help ensure that didn't happen again. So this institute's actually had an interest in climate and its effect on agriculture for many, many years. And I'm also actually an employee. So my salary comes from the taxpayers of the US. I'm an employee of the US Department of Agriculture, which if you jump over this building sits right here. And this campus, I should point out for Tim, I'm sure knows this, is the land grant university of the state of New York. So we have a lot of institutions working on agriculture. And I like to point out, just for fun, the name of our center is the Robert Holly Center. And in this era of genomics, Robert Holly was a scientist at the center in the 50s, 60s, and 70s. And he's the only USDA scientist and probably the only one that ever will win a Nobel Prize. And he won it for actually generating the first nucleic acid sequence, a sequence of a tRNA, 76 nucleotides. It took him 2 years, and he won the Nobel Prize for that in 1968. And in this era of genomics where a student can extract DNA, send it off to a sequencing center, and get 1 billion base pairs of DNA in a couple of days, I like to point this out, that not that long ago, this was very difficult. So our interest, and a lot of what I'll talk about today, is fleshy fruit. And so that fits well within a horticulture talk. We're interested in these fruits who evolution has directed to develop in such a way as where they become succulent, attractive, nutritious, so that organisms, what we refer to as frugivores, will consume them and distribute their seeds. So the fruit have different mechanisms of seed dispersal. It can be through wind, through attaching to animal fur and being dragged away. Fleshy fruits, which we enjoy ourselves and are an important part of our diets, as we heard very nicely much earlier in the day, are our interest. And fruits and vegetables together fall into kind of an interesting class of horticultural products And that compared to a lot of our staples, which are seeds and tubers, have evolved to be very stable. So they can overwinter, they can stay in the ground. They're pretty easy to keep. Fruits and vegetables are very different. Fruits in particular, as part of that maturation process, is a means of making the fruit more susceptible, not just to being consumed by a frugivore, but if something doesn't come along and eat them, you need an escape plan B for the seeds to get out. So they rot, and they're designed to rot. And that becomes a challenge for us in terms of food security and nutritional security, because they do represent a very important part of the nutritional balance of our diets. And when we think about, I think there's also a real opportunity, because we lose so much, particularly in parts of the world where storage and transport systems are not as available. We lose so much of that product that there's a big opportunity to increase net yield without putting more land under cultivation, without doing more, putting more inputs in, just maintaining what we produce and getting it to people who can consume it. And this is important, as everyone that's certainly in this audience knows, we're in a world that's not just challenged by a changing climate, but for probably at least the rest of the century, we're going to continue having an increasing population, probably by about 25 more percent, the projections are to the end of the century than we have right now. So how are we going to feed those people? And being able to get more of what we produce to them is certainly important. So I'll tell you a little bit about fruit ripening. I'll go back to my mispronounced system of tomato. And they do a lot of what most fruits do when they mature and ripen. They convert their starch to sugar, their texture changes, largely due to changes in turgor and changes in cell walls, an area that Graham has worked on for much of his career and made many important contributions to. And those changes are related to these changes in susceptibility to opportunistic pathogens, things that wouldn't normally infect an unripe fruit. So you don't see many rotten unripe fruit. You see a lot of rotten ripe fruit. And that changes texture, pigments accumulate. Chlorophyll is often degraded, not always. Aroma volatiles, the things we smell that attract us, are produced, interestingly, often from necessary nutrients. Breakdown products can become volatile compounds that become part of the characteristic aroma of some fruits. And protective compounds like glycoalkaloids, the things that plants produce to keep things from eating them, are reduced in this tissue as they mature. And we've been interested for many years in what are the regulators, the developmental regulators that control this process, because if we understand them, maybe we can help slow some of the negative consequences down that cause us to lose a lot of the products we produce. So I'll come back to that in a little bit. Before, I just want to, kind of point out how we address this problem now. And we use a number of kind of primary issues, and this goes well beyond tomato to many fruits. We use genetics, so naturally occurring genetic variants or mutations that slow down ripening. We harvest a lot of fruit very early, unripe, immature, and we use a lot of controlled atmosphere and temperature systems to maintain shelf life and quality. Unfortunately, virtually all of these result in incomplete ripening, reduced nutritional quality, and products that consumers often don't like. And I'm sure everyone can appreciate that here. The things you get in the supermarket often just aren't that good. And why is that? And this has been brought up in different ways, I think, in many of the talks, because a lot of our produce is produced in very high intensity, highly efficient production systems, very localized areas, and then distributed over long distances. So obviously this is a map of the US, and just, the tomatoes are produced in California and Florida, but then they go everywhere, and they're coming in from Mexico, from Canada, and even from the Dutch greenhouse systems at certain times of the year. So a lot is done to make the system work, but it often results in poor quality. So a number of attempts have been made, and this is kind of the biotechnology part of the talk. And so I have to say something about GMOs. And I'll give some examples of some horticultural crop GMOs that have come out over the years and just introduce that quickly with something you're probably fairly familiar with, and that is a GMO by definition has a foreign DNA introduced through a biological system, often agrobacterium, or sometimes more direct methods like the particle gun. Some of the cells will acquire that foreign DNA, and then through the presence of other genetic material like antibiotic resistance, you can select for those that have taken up the DNA, and by treating with hormones, eventually regenerate a plant that is now genetically different and will reproduce and carry on that change in subsequent generations. More recently, there's a lot of interest in gene editing, where that's taking advantage of systems like CRISPR-Cas9, which are bacterial systems designed to basically provide immunity to viral infections. And so these are systems that recognize specific DNA sequences and break them. And they've been used and engineered to allow for targeting of specific sequences. And for example, a plant's genome, break the DNA, allow for repair, often causing mutations. And what's interesting is those mutations are essentially indistinguishable from natural mutations. So because of that, at least in the US, these are probably going to be regulated very differently than other GMO type crops. So, of course, probably everyone is familiar with the fact that the first GMO product was a horticultural crop, tomato. The gene was the so-called flavor saver gene, which is a gene involved in that cell wall breakdown that causes textural changes during ripening. Gene's name is polygalacturonase. That work was done here in the UK by Donald Grierson, and also in the US by a company called Calgene. And the whole idea was PG was thought to, well, it's known to break down the pectin between the cells, and that was thought to cause softening. I think Graham and others showed that it's more complicated than that. But, and the idea was, well, then we can make a tomato that can ripen more fully, be of higher quality, but still stay firm so it survives that production system. It was also used, particularly here in the UK, because one of the biggest energy inputs in creating sauce is how viscous that sauce is. And if you start with something that is less viscous, you have to put more energy into it. But by not breaking down the cell walls, the starting material is thicker, so it costs less to produce. And this in particular was on the market for quite a while. These didn't last very long, and we can talk about why that is later if you're interested. But a lot of work's been done in tomato. I just want to point out the reason is because it's an easy crop to work with. It's also the most valuable economically fruit crop in the world, right? So there's more money in tomato than anything else. And I'll come back to that in a couple of minutes in a different context. But there are a lot of good tools that are available that just make it a tractable system. And I'd just like to point out, in terms of GMOs, and this is, I think, sometimes shocking to people who aren't familiar. This is in the US. Many of our major crops are largely GMO. So 90% of our maize is GMO, 90% of our soybean, 90% of our cotton, and so on. Virtually no wheat. Interesting. The wheat farmers never wanted it, so it never happened in wheat. But also notice here, the maize production, corn production in the US is worth $90 billion a year. That's why you see it in these crops and you don't see it in horticultural crops, because it's been estimated the cost to develop and then go through the regulatory compliance of a GMO is about $100 to $150 million. All of the vegetable production in the US is worth $16 billion. All of the fruit production is worth $20 billion. So individual crops in general just aren't worth enough to go through this. So you don't see very many of them. But again, gene editing is probably going to change that, at least in the US. I know in Europe it's a different game. So I just want to spend a few minutes going through some interesting examples of GMO horticultural crops. This is one of the earlier ones known as the rainbow papaya. there was a disease in Hawaiian papaya called papaya spring spot virus that virtually wiped out the crop a number of years ago, more than 90% reduction. This gentleman is Dennis Gonzalez. He worked for the USDA. He made a transgenic papaya expressing a viral protein that essentially inhibits further infection by the virus. The result of that is now that 85% of Hawaiian papaya production is GMO. Production levels currently are exceeding the levels that were at the high point of devastation. And Japan, which notoriously is pretty restrictive on GMOs, is even importing these. This is a small crop grown in one state in the US. It worked because the senator in the state pushed a lot of legislation to let them bypass a lot of the regulatory constraints. Otherwise, it never would have happened economically. You may be familiar with the Arctic apple. This is from a Canadian company that I think has been bought by a US company recently. Here, the gene target is called polyphenol oxidase. It's what causes the browning of many cut tissues. And the idea here was repress it with RNA interference, and you can cut apples and not get the browning. For a fresh apple that you're going to eat whole, that doesn't really matter a whole lot, but for cut products, it's a big deal. And actually, in the US, about 8 million kilograms of Washington state apples are now Arctic apples. And it does have a big impact in these minimally processed products. This is a recent one. I don't know if you have seen these, probably just in pictures. I don't know if they're selling them in Europe. This is the pink pineapple. And it's a complicated GMO. 3 different genes are targeted. Essentially, they're genes that metabolize lycopene, the red pigment in watermelons and tomatoes, that in a pineapple is converted to beta carotene, which is orange, and that's why a pineapple's pretty orange. You block those genes, it backs up in red, and you get this kind of red color in the pineapple. They're really expensive. There's not many are being produced. You pay $10 for one of these if you can find them in the supermarket. You can more easily get them from Amazon. You get 2 or maybe 1 for $30 or $40. So not many people are buying these. But I point this out because these are interesting phenotypes, interesting traits. Most of virtually everything that's in maize and soybean are so-called production traits, right? Disease resistance, herbicide resistance. The consumer doesn't see a benefit directly there, but consumers like these kinds of things, or at least they find them interesting. And certainly a subset of the population is interested in buying them. An example you're probably more familiar with is the purple tomato, developed here in the UK by Kathy Martin. This is overexpression of 2 genes that basically activate the anthocyanin pathway. So there are tomatoes, this is a variety called Cherokee Purple, that naturally has anthocyanin accumulation, but nothing like you see in these lines. And these are now, as I understand, well, as I know, because I bought them, you can get seeds for home garden use. There's going to be fresh market production, processed products this year. And again, Kathy was the one who developed this. And lastly, this is a high GABA tomato. So gamma aminobutyric acid, this is an amino acid. This was developed by a Japanese scientist and Japanese company. It basically targets an enzyme that makes this compound and makes that enzyme more efficient. So you get more of it. And this is a translation of the labeling on products in Japan. And I know you can't see it, I can barely see it here, but it says it lowers your blood pressure, it relieves your stress, it improves your sleep, it makes your skin great and you're beautiful. And we could never advertise like that in the US, and I believe you probably can't here as well without anything substantiating it. But it is a cultural thing as well, because in Japan, a lot of people take GABA supplements. There is an interest in this compound. So these products are very interesting in Japan. So, okay, I'm in no way pushing GMOs. I'm in no way saying they're a bad thing. I'm just telling you what some of these products are. What we do in our lab, and I'll transition a little bit now, is just making sure I have time here, Graham. So, okay. I've spent 30 years asking this question. Ripening happens very fast. We know a lot about it from a hormonal perspective, particularly ethylene. But what causes that very dramatic and rapid change? And so we've been looking at what are the developmental regulators, what are the genetics behind this? Because understanding that process, again, will help us understand how we can make maybe a better quality, longer shelf life fruit. And these are mutants. All of these are naturally occurring mutants that have been identified over the years in tomato. Many of them are bred into the tomato varieties that you purchase in the supermarket. But we and others, and Graham in this case, and my lab in these two cases, in this case, identified the genes that underlie them. And a number of them are regulators, transcription factors. And others are involved, as you might expect, in how the plant responds to ethylene, the hormone that we know is important to ripening, and others are involved in color. And this is Julia Vrebalov, who cloned some of these genes in our lab. And again, a number of them have practical utility. Ripening inhibitor, this mutant doesn't ripen at all, ever. But if you make a hybrid with a normal tomato, you get a long shelf life, slow ripening tomato. This is in most of the varieties you get in the supermarket. And it's one of the main reasons you don't like tomatoes you buy in the supermarket, because they're not fully ripe. But they last a long time. So they survive in that supply chain and in those long shipments. Uniform ripening, this mutation is about 100 years old. And it results in that nice red uniform color. A older variety tomato looks more like this. It has a green shoulder. And in fact, if you're an aficionado of heirloom tomatoes, you look for that green shoulder because that tells you that variety is probably at least 40 or 50 years old. Let's see. Okay. High pigment. This is just an interesting mutation. The plant, it's a gene involved in how the plant perceives light. And the plant believes it's seeing much more light than it is in this mutant. The result is it tries to protect itself by making more pigments. It becomes more red. So that's in some varieties. And I don't know if you're familiar with these. There's varieties from Japan called Kumato tomatoes. And basically, this is a mutation in how the plastids develop. You retain some of your green plastids along with accumulating chromoplasts, the red plastids. And you get this kind of green and red crayons, you'll get brown. And that's the color of these fruit. If you're familiar with what are called chocolate peppers, it's the same exact mutation in those varieties. So these are all bred into commercial varieties. But they demonstrate that if we understand how these things are regulated, we may be able to do something about shelf life and maybe get some of the quality back. So one of the things we've done over the years is ask, well, if it's in tomato, is it anywhere else? Is it unique? Is it conserved through evolution? Most of these regulators are. So we can find equivalent genes. They're expressed in the ripening fruit. And we've worked over the years with a number of collaborators, including Graham, to ask, okay, if you change them in other fruits, do you actually affect ripening? And the short answer is yes, you do. So this is the RIN gene, the equivalent gene repressed in strawberry, and you get delayed ripening or lack of ripening. Same is true in melons and even in bananas. There are two of these genes that if you repress either one of them, the bananas have about 2 weeks longer of usable shelf life. So the biology, you don't reinvent necessarily a lot of new wheels. The same systems are operating a very diverse set of fruits. This is the NOR mutation and just showing, again, the equivalent gene in the strawberries having similar effects. Equivalent gene in kiwi fruit has similar effects. Okay, and this is the most boring slide. I'll just, because it's more recent. We use other approaches, particularly the availability of genome sequence and expression to identify other regulators beyond those that we have in the mutants. A recent one that we call SLAV1, it's a member of this regulatory family of genes called lob-like genes. It's very interesting in that it seems to be involved in controlling just genes in cell wall breakdown. So if you knock it out, the fruit ripened just fine. Ethylene is produced normally. Everything happens the same, but the cell walls don't break down as much. And you get a firmer fruit, kind of what maybe the flavor saver was trying to do originally. This was the work of Giovannoni, a postdoc in the lab. They last longer, they retain their water much better. If you overexpress it, not that you'd want to make the fruit necessarily softer even before it's ripe, but just to demonstrate that's what the gene does, the fruit become softer even before they start ripening. So what we've done and many labs have done over the years is kind of collect all these genes that we know are important. And you don't need to, I don't understand what I put here, but I make the analogy to, it's kind of like having all the parts of a car, you know, and you'd walk out to the parking lot if you came from outer space and you didn't know what that thing was and you just put a knife into the tire and then someone tried to drive it, you would learn that, well, that must be important because now it won't go anywhere. That's what people do. That's what geneticists do all the time. As many of you know, we look for things that break. And then we say, well, that must be important. That tells you what's necessary to make something happen, but not what is sufficient. So what we've done, I think, where we are is we've got a lot of the parts. We don't necessarily know how to put them all together yet. And that's something we're interested in doing. So I'm going to make a transition for the final part of my talk. And I'm showing this slide because I have a student who's a very good artist as well. He made this depiction of the history of tomato. And so the center of origin of tomatoes in South America and the Andes Mountains and down to the coast and on the Galapagos, you can find the wild tomato ancestors. And those were thousands of years ago domesticated into fruits that look much more like what we buy now or grow now in our gardens. It was thought the Aztecs probably distributed those through Central America during their reign. And in the late 15, 1600s, European explorers came over to the New World and brought tomatoes back. Sorry, this was his depiction of Europe right here. And something over there. I apologize. But the point of this, and what's really interesting, is that very narrow germplasm base became the basis of virtually all subsequent tomato breeding. Okay, even what was being bred back in South America and in North America as well. And what that means is there is enormous genetic diversity, and this I know has been touched on earlier, that hasn't been touched, and there's opportunities there to make things better than the way they are. And that's what I'll talk to you about for the rest of the talk. Because it's really just in the last 20 years or so that people have started to understand and use that available genetic diversity. So this is a collection of, down here are the cultivated tomato. These are accessions of tomato that are essentially kind of early domesticates and old varieties, some European varieties from the 1600s and so on. And then the rest of these are the wild species of tomato, except for potato down here. That's just an outgroup. But all the rest of these you can cross to a tomato, which means you can bring genetic diversity from all of these back into the very narrow base that we have of tomato. And again, what's interesting about some of these, even within just these species, you have accessions that are growing on the Galapagos Islands, literally sitting centimeters over salt water, very salt tolerant varieties. Other varieties or accessions that are growing at the tree line in the Andes Mountains, nothing up there but wild tomatoes. So they tolerate very low water, very high UV light, very different climates, and they've evolved to adapt to these environments. So all of those are opportunities. And what I won't show you, but even in some of these very, weird little tomatoes, and all of these, including the closest relatives to the cultivated tomato, are just tiny little fruit. There's nothing in the wild. Someone mentioned that earlier. There's nothing in the wild that looks like a cultivated tomato that's gone through domestication and breeding. So, but even from these, you can bring in, we find important, useful traits for the fruit, things you wouldn't expect just looking at the fruit, because there's just a lot of genetic variation. So, and about 10 years ago, with the sequencing of the tomato genome that both Graham and I were involved in, we now have access to what is in these genomes. And more recently, this is just a graph of how many tomato genomes have been sequenced in this, 11-year or so time frame. And now we're up to 75. This is fully sequenced from scratch genomes of the different species as well. And there's actually over 1,000 genome sequences from tomato from just different cultivars, not to this quality, but there's a huge amount of information that we can access to just see how those genomes are different in all these accessions that do different things. Okay, and finally, in terms of the real boring stuff, advances in how DNA sequencing is being able to be done very efficiently, and more recently, just in the last couple of years, very long sequences lets us capture a lot of variation that was lost even in the initial genome sequence. So we really have a good view of how these genomes vary. And so what we're doing is we're taking many of the modern tomato, many of the old tomato accessions, and then the closest wild relatives, the things from which the modern tomato was derived. And we're making sure we have a collection, so now of about 200 accessions that represent as much as possible of that diversity within those species. And there's a few where we're sequencing them ourselves because they're missing. But so we have this diversity collection. And then we've been collecting a lot of gene expression data so we can say, okay, here's how the genome changes. Here's how that influences expression of genes. Okay, that's nice, but so what? Well, we can then start seeing, we can go backwards and see some of the interesting things that have happened. This is a gene called CCD1. It breaks down carotenoids, okay? So if you have high expression of this gene, you actually have low carotenoid levels. And if we just look at how the gene is expressed in all these different genotypes, what we find is actually a lot of the wild species, the ones that have less carotenoids, have high expression of the gene. The cultivated tomato has very low expression of the gene because breeders have selected not for the gene, but for higher color. So again, what we're trying to do is set up a system where we can capture a lot of genetic diversity, including from the wild ancestors, characterize a lot of characteristics of the plant, in our case, particularly the fruit, layer on understanding of expression of genes so that we can go through that diversity and ask, can we associate changes in the genome that have evolved over time under the best selection pressure there is, environments that are very different and that have changed over millennia, for differences that influence things that we're interested in, like stress tolerance. I put that in here because I know this audience would probably appreciate that, and fruit quality traits. So I think I'll just stop. I won't even read the summary since I'm short on time, but thank you for your attention. If there's time for a question, I'm happy.